Hello everyone. Today we're going to discuss platelet aggregation in hemostasis. So we've got here in this graph blood vessel as it is. So we'll just start naming some of the bits so that we know where we're at. So this at the very bottom where the lumen of the blood vessel is your tunica intima. This middle layer here, where you got the smooth muscle cells and the collagen, this will be your tunica media. And at the very top, where you've got all this yellow scaffolding structure, when is your collagen, and these blue smudges here represent your fibroblasts. This is the outer layer of your blood vessel and represent your tunica adventitia. Here we go. So that is where we're going to start and work from. So our tunica intima, as we said, is lined with endothelial cells. And your tunica media, so these pink structures here, are your smooth muscle cells involved in the contraction and relaxation of your blood vessels. So involved in your vasoconstriction or vasodilation, which is your smooth muscle cells. And as I said before, this yellow sort of a structure here is like a scaffolding protein that serves to give a better structure to your blood vessels and to all sorts of tissues really in our body, and is called collagen. And you will find some in between your tunica intima and your tunica media, and you will find a lot of it in your tunica adventitia, so you'll find some in here as well. So that's where we're at with our blood vessel. So what else do we have free floating in our blood vessel? Well, we said that in a blood vessel we will have our platelets. Yeah, so we'll draw them here, as you know. Here we go. So we'll have free floating platelets, which are often what they do, they stick on the peripheries of your blood vessels, almost as to guard the integrity of your endothelium. Yeah, so we'll mark these as platelets. Here we go. Something else that you find free floating in your bloodstream is a very important glycoprotein, which is called the von Willebrand factor. And von Willebrand factor is a much smaller glycoprotein that is a bit free floating a little bit all over the place around here. Okay. Von Willebrand factor, we're just going to abbreviate it with VWF. This is von Willebrand factor. And on top of our platelets, so let's have a proper good look at them here, you do have some receptors, in green as well. Here we go. Some receptors like this on all of them. That's it. And these receptors are actually, they are von Willebrand factor receptors. But the problem with it is that they can't bind, but your free-flowing von Willebrand factor cannot bind on these receptors in their current form. In the current state, they need to undergo a certain conformational change before they can actually bind to these receptors. Now, we also mentioned that platelets like this in this current state, they are not gonna coagulate, they're not gonna aggregate this way. So this is because of what? Because my endothelial cells here, they constitutively express some structures, some chemicals which stop platelet aggregation, don't they? And these are specifically nitric oxide, or actually the chemical formula would be simply NO, nitric oxide, and prostacyclin. And nitric oxide and prostacyclin, what they do, they ensure that the blood stays nice and fluid and it doesn't start to coagulate in its natural normal form. So nitric oxide and prostacyclin, also known as prostaglandin 12, are constitutively expressed by the endothelial cells, by the endothelium, constantly. Something else that helps with keeping my blood fluid and uncoagulated in my normal state is, we'll draw it here, is physiological heparin, but it's not as important for this now because physiological heparin, but we're going to put it here, is present in the membrane of the endothelial cells. So here, just going to put it here like, here you go. Cool. Here we go. Heparin, here we go, on the membrane, following the membrane of the endothelial cells, but they don't really act at all on the platelets. And what they do instead, they cleave to some clotting factors like on thrombin and factor 10A, and they make them inactive so that the coagulation cascade won't start. So this is my blood vessel on a normal state. Now, what happens when my blood vessel gets damaged? Here we go. So let's have a look at the damage here. Well, the first thing that would happen, you have my endothelial cells will start secreting what we call endothelin. Now, endothelin, so I'm just going to put it here, this substance. Here you go, this stuff here. What it does, it's secreted by damaged endothelial cells, and this is a peptide that causes vasoconstriction. So as a result of this damage, so I'm going to put it here, vascular damage. Here we go, just right there. So that's what happened here, right? So what happens as a result for endothelin being secreted by my damaged endothelial cells? What happens? Vasoconstriction happens. Here we go. And this is what we call that is my initial response to hemostasis, which is vascular spasm. Now, what we are mostly interested in now is the formation of my plated plug. So, what happens here is quite interesting. So, but we need to have a proper look here at the damage. The first thing that we notice is that all this collagen that is in between my endothelium and my tunica media and my smooth muscle cells gets exposed to the blood, is it gets exposed to the lumen of my blood vessel, which usually doesn't happen. So what will happen? What will happen then? All the von Willebrand factor that was just free flowing in my bloodstream. So let's not forget this is von Willebrand factor, will start binding to this collagen. You see, we'll move it from here. Here we go. And what it does, it will start binding. Here we go. To all this collagen. And it's going to make sure at this point that its shape will change in a way that is now able to bind to, you get it absolutely right, the von Willebrand receptor on my platelets. So let's have another even closer look to my damage here. Here we go. So here, there we go. So what's going to happen? My von Willebrand factor has now bound to collagen. The small conformational change has happened. So this will allow my platelets, here we go, so my platelets, to bind to it. Just like that. Got another platelet here. Here we go. Another one there. Here we go. So at this point, all my platelets, they start binding. They're starting to conglomerate here at the site of my vascular injury. So what happens when platelets bind to my von Willebrand factor and my site of injury? Well, what happens here is a process called platelet activation. Now let's have a look at platelet activation, shall we? So what's going to happen during platelet activation? So there's a few things that happen at this point. The first thing that happens is that my platelet, as you can see here, it undergoes as well a significant conformational change. And in order to extend its surface area as much as possible, it develops these tentacles as such, which are called Philippodia. And the development of Philippodia is to extend the surface area of the cell so it can cover a bigger area. And also, they will allow to bind to more von Willebrand factor and collagen. So the first thing that happens in platelet activation is a conformational change. Conformational change. Here we go. Development of Philippodia. Another thing that happens here on the surface of my platelet is this. There is other receptors, which were always there, of course, but now they get activated on the surface of my platelet. And these are called my fibrinogen receptors. Go. And fibrinogen receptors, they get activated once my platelet cell is activated. So the second thing that happens during platelet activation is activation of fibrinogen receptors. Now, why is this so important to have these fibrinogen receptors? So fibrinogen, it is also just free flowing in your bloodstream, and you will see it has a massive importance in your clotting cascade, which we will see later on. But also in this case, it helps forming a bridge in between platelets in order to start forming this plug. So let's have another look like in here. So if we have a look, a proper good look in here, we have this fibrinogen receptors, and actually, you know what, we might have this platelet that has generated some Philippodia like that. Here we go, it's expanding as much as possible. Here we go, like that. That's it. And obviously, fibrinogen will then try to bind to it, yeah? So this will be my fibrinogen. It will allow for what? Will allow for other platelets, say so here, to bind to it this way. So this is another platelet has formed, Philippodia and all the rest. This way, just like that. So they will bind to each other, forming what is called a fibrinogen bridge. And this is how all the platelets, they start sort of getting all together and get attached and start stick to each other thanks to this fibrinogen bridge. So back to our platelet, where was it? Here. So we said the first thing that happens is a conformational change in the development of Philippodia in order to extend their surface area as much as possible. To be fair, almost simultaneously, well, we'll put it as a number 2, what happens is the activation of fibrinogen receptors so that fibrinogen can bind to it and then form this fibrinogen bridge that allows the platelets to sort of start sticking to each other, which giving sort of a rudimental form of my platelet plug. But the third thing that happens, which is quite interesting, is is my platelet degranulation. What does that mean? Well, simply, inside my platelets, we have some sort of, we'll call them sacs as such, okay? And these sacs, okay, we'll put them here, they contain some granules. And some of them, so we'll call this a different color, here we go, pink. Call it that. Here we go. There's different types. These ones, they're called the alpha granules. And there's some different kinds here, which are called the dense granules. So when the platelet degranulates, what does that mean? It means that when it gets to this point, here we go, it starts releasing all of these structures. So it starts releasing my alpha granules and it starts release my dense granules. Here we go. This is my degranulation. Oops. And this is my Now, what are these alpha granules and what are these dense granules, and are they the only thing that matters? They're actually quite important. So the alpha granules are, well, this will shock you, more von Willebrand factor, so that if there's more collagen exposed, there's more von Willebrand factor gets activated, so that more platelets can bind to it, and it will also release fibrinogen. So that we can keep on creating this fibrinogen bridge in order to get more and more platelets to stick to each other. The dense granules here we go. This is once I read a very interesting way to remember what is inside the dense granules, because they're like sacs. Yeah. Here we go. They're like a sac. This will help us remember that they release serotonin, which also helps with vasoconstriction. ADP, adenosine diphosphate, which it does, helps with the activation of more platelets. And then calcium. And calcium will play a really important role in your full clotting cascade and the activation of different factors in your clotting cascade and in the strengthening of your final clot. Now, another thing, though, that gets released from the platelets, it's something that is not contained in a sac per se, but is in the cytoplasm. And what is that? And what the platelet cytoplasm release is another compound, and this compound, what it does, it helps with the recruitment of more platelets, and this is thromboxane 2. we'll call it recruitment. And at this point, you might think, why is this in any way relevant to my paramedic practice? Well, aside the fact that it's an important process to know, this thromboxane 2 and the recruitment of more platelets, this is where your clopidogrel acts on. So clopidogrel interferes with thromboxane 2 and stops or slows down, really, the recruitment of more platelets, hence helping with the blood not to coagulate as much. But it's not so much the coagulation that affects, is the recruitment of more platelets. And within the dense granules, your ADP, which what it does, it gets secreted by the platelets and it starts activating all the different platelets that are around, which means making more platelets degranulate, more platelets developing philipodia, more platelets just activating those fibrinogen receptors and creating bridges. And this is where your aspirin works on. So when we say that aspirin helps thinning out your blood, well, really is an antiplatelet that acts at your ADP receptors, are your adenine-diphosphate receptors of your platelets, and stops more platelets being activated. And this is how the platelet aggregation works.